You're going to wear your hat for the introduction?
Should I not? You know, should I not?
That's fine.
That's fine. I could also change.
No, that's fine.
You know, this is a typical like what do you call it? A French panties, panties cap, or put a mustache. That's also fine. Or why don't you play around? Why don't you if you like to ski? Huh?
Okay. All right. Okay.
That's too much. I'll just put this one just for fun. But why don't you join us? It will be fun.
I don't have the latest version of Zoom that does that, as far as I know.
All right. You have about a month to try that new feature before the next ZOOMinar.
Okay. Yeah. Oh, that's right, Jen. You should announce that we're.
Yeah, I'm gonna tell them. Yeah.
Yeah. So it's January 12th.
Exactly, Bob. January 12th.
Yeah. Okay.
Mm-hmm.
You need to change your background too, Kong.
Okay. Just give me a few seconds.
You should have an Eiffel Tower behind you or something.
Exactly.
Okay, I will do that right now.
You know, the Arch de Triomphe.
It's called Up to Tomb.
Champs-Élysées, a picture of Biden.
So, Bob, if we were on TV, people would be looking at the titles of the books in your bookcase.
We're looking at the titles of the
books in your bookcase behind you.
Okay, right. Well, it's a little bit hard that they're somewhat small to read, but it's only quantum mechanics books. It's not very interesting. There you go, Kong.
Yeah, but it's not very good. It's like I'm blocking.
That's a.
It looks like it's coming out of your head.
It's not very good. I need to find.
You need to shift it to the left or to the right.
Yeah, it is better.
Very nice. Perfect.
That's much better. That's much better. Right.
Yeah. I'm in LA now, Las Vegas. No, LA. Just joking. Thanks, Jeffrey. Thanks for your approval.
Okay, we generally wait till 11:05, which is sort of MIT time to get started. We have another minute or so, and then we will get the show on the road. All right. Okay, Kong, why don't we go? All right.
Yeah, just give me a
Yeah.
minute. Let me try to pull up the things that I usually say. Hello, everyone. Greetings again, and welcome back to our Solid-State NMR or DMP ZOOMinar series. I mean, it has been quite honor, like being able to do all this for almost like half a year now, since the start of our first series in April. And now, like we would just like to announce that this the session that we have today is the last one of this year. So, and we're gonna start again in January 12th. So thanks for all the support, and also like I mean like we are happy to do this, and of course it's not just us. There are a lot of like other people. Of course, Mei Hong is on another series doing the bio NMR series, and then like apart besides the one organized by Mei and Göttingen people, they have also been other like groups doing the same thing, like similar things, and in fact so Lucio wrote a very nice article on JMR, like talking about like all these virtual series and how this virtual series try to keep everyone together at these like difficult times. So I mean, if you're interested, feel free to look for this article. And I mean, of course, I do not hope that the pandemic will continue, but we hope that we can still able to do this for the in the future. I mean, in January. So stay tuned. So I will do the usual thing. I already put up the mailing list, the recordings, and the websites, and the Q&A, the instruction on it for it in the chat room. So apart from that, I think there's nothing else for me to say. So Bob, would you like to introduce our speakers today?
Okay, yeah. So I guess the first thing that everybody should notice is that Kong has graduated from MIT, all right, and moved to Paris, where he's now a junior professor in the laboratory for biomolecular NMR. Is that what I forgot the exact title of it? At the École Normale, where Jeffrey Bodenhausen and Daniel and so forth, the big group of NMR people are. So unfortunately, MIT has lost him, which is a big loss for us. But in any case, today we have two very exciting talks focused on membrane proteins. And the first one is by Mei Hong, who's a professor here at MIT. Mei is a graduate of Mount Holyoke College, and she got her PhD at the University of California at Berkeley. She was a postdoc around here at the Magnet Lab for a year or so, and then a professor at Iowa State for a number of years before she moved back to MIT, 2014 or so. And today, Mei is going to talk about ligand-bound structures of membrane transporters using fluorine-19 and proton solid-state NMR. So Mei, the virtual floor is yours, okay?
Thank you, Bob. So, all right, I think. All right, there's a, oops, something like a 4-second darkness. You guys are seeing my screen okay, or?
It's okay now, yeah.
Okay. Thank you, Bob and Con, for the invitation to speak at this very successful ZOOMinar. It's kind of funny. I've hosted a bunch of seminars by other people, and I don't, I don't have a chance to talk about my own thing so often during this pandemic period. But I'm very excited to share a new study from my lab to understand the ligand-bound structure of a bacterial transporter. We determined the structure using proton-detected fluorine NMR. And so this is exciting to us both because of the biological significance of this bacterial transporter and because we're able to apply the fluorine NMR techniques that my lab has been focusing on in the last few years. Fluorine NMR has a pretty long history, distinguished history in both solution and solid state. And many workers have dived pretty deep into this. In solid state NMR, Jake Schaefer is one of the forerunners, and Wurge has applied it to oriented membrane work. And recently, Tatiana Polinova, Anne McDermott, Chad Ringstra, and Lyndon Emsley have all done quite a lot of work, MAS fluorine NMR. And in solution state listed here, you see, many people apply fluorine to NMR to large proteins or membrane proteins, and recently Hari Athanari and Gerhard Wagner have done nice work developing fluorine-Chose NMR. And the reason fluorine is really nice to NMR spectroscopists is because of its natural 100% abundance. It's a very nice, convenient spin 1/2 nucleus. It has no background in biological molecules, and yet at the same time is really pretty easy to incorporate into proteins, both recombinantly by simple methods like glyphosate, and also synthetically into peptides. And they're organic chemists who developed carbon-fluorine bond formation techniques. So partly as a result of that, about 30% of pharmaceutical compounds already contain fluorine, so you don't have to do anything special to get these. Fluorine chemical shift is really sensitive to structure, chemical structure, three-dimensional structure. And so even just with simple 1D fluorine NMR, you can already tell a lot about the protein conformation change in response to ligand binding, for example. Now, the property that I'm particularly interested in is the high gyromagnetic ratio, or the magnetic dipole moment of fluorine. It's only 6% smaller than proton. So at a 600 megahertz machine, you're getting 564 megahertz Larmor frequency. And this high gamma means you can measure distances to further than compared to C13 or N15 low gamma nuclei. So if you look at a 30 hertz dipolar coupling, if you think about homonuclear fluorine-fluorine, then you can go out to about 1.5 nanometer compared to carbon-carbon 6.3 angstrom. But here in this talk, I want to particularly focus on heteronuclear fluorine-based measurements, carbon fluorine as one ruler and proton fluorine as another ruler. And so here I just want to show you the kind of fluorine chemical shift span or dispersion that you can get in small organic compounds containing aromatic fluorine, such as this one, PNC shorthand, or sitagliptin, which is an anti-diabetic drug with trifluoromethyl, and these aromatic fluorines resolved here. And then we incorporated fluorine into the tripeptide model compound MLF, and the chemical shift spectrum is here. And we expressed tyrosine triple fluorinated GB1 without too much sweat. And you can resolve these three tyrosine fluorine signals. So all of these spectra were measured at 600 megahertz using a 1.9 millimeter HFX probe. So you can simultaneously tune to the proton and fluorine channels, so you can decouple. We spun these samples at anywhere between 25 and 40 kilohertz to get rid of the sidebands, or at least make them not cost you too much center band intensities. We also have in the lab a 1.3 millimeter shared HF channel HFX probe. So you can have both channels at the same time, but if you get to 50 or 60 kilohertz MAS, you can actually get away doing fluorine detection without proton decoupling. So this gives you an idea of the fluorine shift dispersion. Now, the interesting techniques that we want to explore are the kind of multiplexed heteronuclear read or base distance measurements. And here I want to emphasize that we really want to measure a bunch of distances in one go and not just one at a time as the typical traditional 1D read or experiment would be. And to do that, we need to incorporate this type of heteronuclear dipolar recoupling module into the template of 2D or perhaps 3D correlation experiments. And so here I have a 2D CC experiment done under a spinning speed that's possible for a 1.9 millimeter probe. So we put RFDR here as our 2D correlation scheme. Spin diffusion still works to some extent, but the important point is we put a carbon fluorine module in front of before the T1 evolution. So you're looking at fluorine dephasing of the omega-1 dimension, F1 dimension carbon nucleus. And now the first question you might ask is whether ReadOR still works well for this kind of spinning speed 25 to 40 for fluorine CSA that is anywhere between about 20 to 50 kilohertz. And the simulations here show that if you only spin at 12-ish kilohertz, then that's no good unless you have only CF3 group, a really small CSA, because your readout curve, for example, for the green one, 50 kilohertz CSA is no longer, is pretty far from universal. You can still simulate it, but it's not very predictive. And if you spin up to 50 kilohertz, then the curves become nearly universal, pretty close. And so here I can generate a readout panel, simulated panel for different distances for a reasonable combination, a 30 kilohertz, MAS, I can't wave this, and 40 kilohertz, 30 kilohertz CSA and 40 kilohertz MAS. So this is the approach we take. We do the 2D CC correlation, and here I just show you the C alpha to carbonyl region of the GB1 spectrum containing 3 fluorinated tyrosine residues. Again, the sample came from expression, and we just apply glyphosate before turning on the protein expression, and you feed the bacteria with the fluorinated tyrosine. That's all it takes. And so we take a pair of 2D spectra, one control without the fluorine pulses, and then the other one is dephased. And in a different spectrum, you see all the carbon signals that come from those carbons whose C alpha is close enough to the fluorine to show a different signal. And so you can dial different mixing times. Here, this is 8.4 millisecond. If you use longer, you get more peaks showing up. If you use shorter, then it's all the closest carbons. And so this is the strategy, just the 2D extension of 1D readout. And here, the readout curves are shown as a S/0 dephasing. Just with 2 mixing times already can get you down with a distance precision better than 0.4 angstrom or so. So these are the different C alpha groups resolved in the 2D CC spectrum. And you might ask, okay, which fluorinated tyrosine are you dephased by? Well, in this particular case, when we did these experiments, we didn't resolve the fluorine signals. Later, we show in an experiment, you can actually do carbon-fluorine correlation. Then you can really pick out, which fluorine is doing the job. But even with this pretty significant approximation, we just said, okay, we're going to take the nearest tyrosine in the structure, in the known structure GB1. Let's just see how good these distance distance analysis would be. We assume a single spin pair, which is again a significant approximation, but that's good enough actually for us to get this particular distance correlation panel where the x-axis is the measured experimental distance, assuming a single readout dephasing, and y-axis is the benchmark gold standard PDB distance. And the precision or accuracy is about 10%, meaning when you go out to about a nanometer, you have inaccuracy of about 1 angstrom. So later, when we went to a different sample using a singly fluorinated tryptophan, then this should be better. This is better than this triple fluorinated system, just because you could have a carbon that's in the dipolar field of more than 1 fluorine. So now what we really want to get to actually is this proton detected version of readout. Now here the proton comes in 2 ways. You want proton detection to enhance the sensitivity. So you want a deuterated protein with back exchange. And then we want proton fluorine readout. So the 2D readout is the NH HSQC type of experiment. And we insert the readout right before the proton detection. So that's all it is. And here, the demonstration comes out even more accurate as well as nicer quality spectra. So this is CDN labeled GB1. You can see the S0 spectrum for one of the mixing times. And we do this again, a pair S0, S for each mixing time. And then here, I just want to show the delta S for one of the shorter mixing times, 1.8 millisecond. You see only a few spots of those amide hydrogens that are close enough to that single tryptophan fluorine. And then if you go out longer, you get more spots showing up. And here I have the S/0 dephasing curves for the various resolved amide hydrogens. So now you might say, all right, this is if I take 6 mixing times, then how long does that take? Well, each 2D only takes 3 hours. So I have a pair of 2D for each mixing time. And so 6 experiments, it still takes only about 36 hours. So it's really fast. You measure out to long distances, and the precision is good. So here, the precision, I would say, is about ±1 angstrom around that gold standard diagonal. And here gives you a sense of the kind of distance we're measuring. So you can actually calculate this GB1 structure. We did that using these distances and the assigned chemical shifts, which you have to do anyway. So let me show you what we can do with this technique. So after I talked about this work in ENC, last year in 2019, Katie Hensler-Waldman came to me and said, well, can you do something about this, EMRE? And I've been very interested in that field, this beautiful work done by Katie, as well as Nate Treseth, and also to some extent from Clemens Galbit's group. This is a very interesting drug transporter. It is a antiport, as far as we know, until very recently, it's a secondary active transporter of bacteria, which means what it does is that it pumps out drugs, and these tend to be polyaromatic cationic drugs, out of the cytoplasm of bacteria into the periplasm. And it does this against the concentration gradient of the drug, because you want to apply antibiotics, right? So you have more drugs on the outside than inside, and these transporters will defeat the antibiotics by pumping out the drugs. And so EMRE, just like its family of this SMR, small multi-drug resistant family of proteins, can do this only in the presence of a proton concentration gradient. So most of the literature says, well, the drug export is coupled to the proton import, although there's recent data that says that this direction is not necessarily opposite, that it can also be symport. But anyways, I'm gonna go by the traditional model of the antiport. And so, all right, so this drug, the drug that's exported is aromatic rich, and it's cationic. And so a lot of pretty extensive literature contributed significantly by Katie Hensler-Waldman's lab says that this transport proceeds in this alternating access model mode, where this dimeric protein, MRE is dimeric, 2 subunits, alternate between an outward facing, outward open, and inward facing, inward open conformation. Initially, you might think of the outward facing ready to receive 2 protons. So 1 proton per subunit binding, to a glutamic acid residue that's ready to be protonated. And then binding the 2 protons would convert the protein into the inward facing conformation, releasing those 2 protons, and then allowing the drug, cationic drug, to bind from the inside, and then convert again to the outward facing mode, releasing the drug, and then the process starts again. Okay, so that's the model. And what's fascinating about this protein is that it is not just a dimer, it's an asymmetric dimer, which means the 2 conformations are not the same. You get 2 sets of chemical shifts for this 110 residue protein. The 2 subunits go in an anti-parallel fashion, and that's pretty unusual. And it took a crystal structure in 2007 to really nail this. It was somewhat controversial, but the crystal structure in 2007, obtaining 3.8 angstrom resolution for the backbone, showed this relatively conclusively. And then this was confirmed later by other biophysical methods. But this resolution is not high enough, and there's sidechain information is not there. The rough, drug binding position is known, but it took another 10 years for Karpla's lab to do MD simulations to fill in the sidechain conformation. So again, no experimental input on the sidechain. And of course, the sidechain is what's important for binding the substrate. And so, so there's a lot of interesting questions about this transporter. Why is it promiscuous? Just how does it, take the structure and convert between this outward facing and inward facing? And maybe are there more intermediate states? So lots of questions, both from the structural and dynamic side. So, so what Katie mentions is that they actually have a classically studied drug, TPP, tetraphenyl phosphonium. So there's a phosphorus in the middle, and then otherwise a tetrahedral drug. And they had a fluorinated version of this obtained from University of Alabama from Jim Davis' lab. And so this fluorinated drug actually also gets transported by the protein. And the solution NMR data from her lab shows that the fluorination versus the original hydrogenated version doesn't make much difference to the protein conformation, because if you look at the Mi proton chemical shifts and nitrogen, the difference is very small due to this fluorination, and that difference is small compared to the intrinsic chemical shift difference between monomer A and B anyway, as shown here. All right, so no problem incorporating the fluorinated drug. And they also did transport assays to show that this fluorinated drug is transported by the wild type protein. So focus on the filled red bars here. I want to just explain this a little bit because it's interesting. If you apply a inward facing proton gradient, but no drug gradient, there is proton current. That's what they measure, the cation current, and that goes into the positive current means proton in, cation in. And if you have an outward facing drug against the proton current, then this inward current goes higher because the outward, drug current makes this proton go in further. Now, if you apply the inward facing drug gradient, then the overall cation current goes in the opposite direction because that drug gradient favors the proton going out. And that's what they measure predominantly. So basically this transport assay says that yes, the fluorinated drug is transported. So Alex Shubakov in my lab took a look at the fluorine NMR signature of this drug, and it's pretty interesting. In low temperature, we see a near rigid limit CSA, but if you go to the physiological temperature, this becomes isotropic, and it's most likely this tetrahedral drug is undergoing tetrahedral jump. Now, in the intermediate temperatures, what we're interested in, because well, here you can actually see a fairly heterogeneous line shape. And if you deconvolve this, you can see a 6% component, which has really narrow lines. And that component gets preferentially enhanced if you do carbon to fluorine CP, double quantum CP from protein to drug, which means that this component is well ordered, is really close to the protein. This dynamics at high temperature is coupled to the protein dynamics. So there's a little bit of protein motion that goes with it. So this gives you an idea of the inherent dynamics of the system. So I just want to point out this EMRE contains a mutation to facilitate the spectroscopy. So now we're ready to apply our proton fluorine methodology, and just to show the pulse sequence again. So we have the CDN labeled protein that we're measuring these HSQC type of spectra. Here's a S0 spectrum, and I don't show the S because the delta S is the more important one. And if you go to 1.68 millisecond, you already have various peaks, difference peaks showing up. These are the MI hydrogens that are close to the drug. And if you go out longer, more peaks showing up. And we can resolve all of these from 3D experiments that I will show you in the next slide. All of these are done at 38 kilohertz MAS at moderate temperature. And you can see how these dephasing, redoor dephasing curves very nicely tells you distances to some of the drug proximal residues, such as the glutamic acid 14, which is known to be the proton binding residue. And there's a tryptophan 63 that's really short, 5.8 angstrom. This comes from actually the sidechain indole nitrogen. And so this is consistent with biochemical data that's already known that the tryptophan is important. We also have some long distances from, that sites that dephase just a little bit. So how do we resolve all these signals? We did a bunch of a set of 4 3D experiments, correlation experiments. Alex is the one that did most of these, and Shiva Mandela in my group helped with the resonance assignment. And you can see how, taking this kind of experiments that are championed by Guido Pintacuda and others, we can walk through the backbone, assign all the proton, Mi proton, nitrogen, C alpha, a little bit of C beta, chemical shifts, and you can walk down from the intra-residue HCA NH to inter-residue HCACO NH, as you can see in this way. And if you look at the carbonyl region, you have two more experiments to take care of that. So at the end of the day, we can get all the chemical shifts combining with the NCC type of experiments. We know the secondary structure of the protein well. We know where the loops are between the 4 transmembrane helices. So I forgot to say this protein has contained 4 transmembrane helices, each copy. So you have 8 altogether. And this is a busy diagram because I want to show the short distances that we observed in the HSQC spectrum. These are shown in these pink spots. And so they are populated or clustered in the transmembrane helix 3 and 1. Now here I already have to show you some simplified diagram of the protein structure because this is so complicated that if you don't have it to guide yourself, it's hard to see. The drug is in between the 2 monomers at the interface between the A and B subunit. So in orange is A, and in slate blue is B, and the drug is in between, and it's kind of surrounded by helix, transmembrane helix 3. Oops. All right, stop jumping. And transmembrane helix 1. All right. So now what can we do to actually calculate the structure? taking all this fairly complicated information, bunch of information. So what Alex did in my lab was to take the chemical shift in the typical way, Talos, bring it into GROMACS, but importantly, we would like to have these amide proton to fluorine distances to constrain the structure. Now, we first have to dock this substrate into the dimeric apo structure that's known, low resolution, and the docking is done in haddock. Now you look at this drug, it has 4 copies of fluorine. We measured 72 unique dipolar couplings. Which of these 4 fluorines dephased needs to be sorted out? And that can be sorted out once the docking is achieved. And this haddock gave us a well-clustered single position for where the drug is. And from that, we can do a structurally based HF assignment. Not every dipolar coupling corresponds to a unique HF pair. Some of these couplings have similar enough distances that we have to allow multiple fluorines from each drug to dephase the proton, and that's okay. So we, in the end, obtain something like 200 or so protein drug distances. And then we take that docked result done where the protein is held rigid and bring it into GROMACS to do MD simulations so we can relax the protein backbone conformation. All right, so the starting point is the carpless modified or carpless refined crystal structure, APO, and then we dock with the protein protein drug distance constraints, and then we equilibrate the structure. So here comes the MD simulated or refined, GROMACS refined structure constrained by all these distances, as well as the device side torsion angles from chemical shifts. I want to show you, so here comes a bit of a closer view of the binding site. So I want to emphasize the E14 residue. So you see that the drug is bound in an asymmetric fashion between the 2 E14 residues. And this is important because this transporter has asymmetric pKa. So the drug is closer to copy A, subunit A, E14 than to B, suggesting, because this drug is cationic, that this E14A is anionic, while that one is neutral. And that would be consistent with the pKa measurements from Katie Hensler-Waldman's lab. We also see the pair of tryptophan 63 residues here, and then the other aromatic pairs. So if you combine all of this, you get this very busy binding site view, which is hard to see without turning it around in pi-mole. But I want to convey a few points that this binding site is relatively open and spacious. And so I think it's a lot of weak aromatic interactions that stabilize together the drug, but the fact that it's spacious would explain why this protein is promiscuous in binding many things. The asymmetric binding is important for explaining the two different pKa's that's known from previous work. So I want to compare how the NMR structure differs from the carpless simulation prediction. You see how overall, I mean, the drug TPP is roughly in the same spot, but the orientation of the 4 phenylalanine ring is different. The relative displacement of the 2 copies of these key aromatic residues is different. The E14's proximity is different. And so just a lot of details are different. So in conclusion, I hope I have given you a sense of the biophysical insight that we can get from this high resolution structure determination using this fluorine proton reador method. And this kind of technology, I think, is really very complementary to the carbon M15 and proton-based 3D experiments that a lot of us are doing. Using the similar type of approach, carbon fluorine in this case, my lab also just solved the transmembrane domain structure of the SARS-CoV-2 envelope protein, and I will talk about that on Thursday next week in the Emerging Topics seminar. Welcome you to come to hear that. So with that, I'd like to really thank Alex Shcherbakov, who's really instrumental, the leading person, to conduct this extremely sophisticated project with lots of computation and spectroscopy, and Shiva for a lot of brainstorming and helping with resonance assignment, and my collaborator, Katie Hensler-Waldman's lab, and also the drug came from Jim Davis' lab in Alabama. Thank you for your attention.
All right, thank you, Mei, for your very, very interesting talk, and like thanks for showing us the development of the 19F readout spectroscopy and using it to for the structure elucidation of the TPP drug to the EMRE protein. Just before like we read the question, so I would like to invite the audience to type out your questions in Q&A, and I will just like give you a few minutes to do that. While doing that, let me just if I may, I would like to ask a very, very simple, quick question. So I'm just curious. So some of the data that I'm not sure, like, well, some of these like fast experiments performed using a 1.9 or 1.3 probe, because I think the MS frequency were like 40 or 38 kilohertz. Well, why not spins faster if you use a 1.3, or were you using a 1.9, or I'm just curious?
Yeah, we're limited by the probe technology. We don't happen to have an HFX 1.3 millimeter probe. I think these experiments will go actually a little better and faster if we can have a 1.3. So right now we're using the 1.3 to do the standard HCN experiments. We just cannot quite have separate proton fluorine channels. Now, Tatiana's lab has shown that you can actually kind of sometimes get away, just not worry about proton decoupling if all you want to do is fluorine detection and doing something on the carbon side. In this case, our method is proton detected with fluorine dephase. So I really cannot, sacrifice. Anyways, we're limited by the probe. Yeah, I've been hoping Booker will extend. I think they can do it. It's just they, you have to keep pushing them to deliver a HFX probe at fastest speed. Yeah.
All right. I mean, like that would be very, very interesting to see if it can spin 60 kilohertz, like getting like much better proton detected, higher resolutions. So
yeah, exactly. Yeah. Right. 40 is a little bit slow. 40 is a little bit slow, even with deuterated. Yeah. So.
Yeah. Okay. Thanks. So let's read. So let's look at the first question asked by Vod. I mean, our good friend at MIT. In a case like this, can you do anything useful with the 31 phosphorus nucleus?
Yes, phosphorus NMR is also very useful. In fact, that phosphonium, I mean, the middle phosphorus has been used by Clemens Glaubitz to measure, and I think it might be a distance measurement, I'm not sure. But so as long as you have a multinuclear probe, sure. I think though that fluorine sticks out and it's much, much closer to the protein. So it's really nice. And it's also very sensitive to the environment and so on. Yeah, so in my lab, my phosphorus probes are at low field, 400 megahertz. And I think I don't have a phosphorus probe for fast spinning and this type of application. Yeah, so this otherwise, absolutely, it's a beautiful nucleus, yeah.
Yeah, but I don't know, I'm not sure, but I also think that phosphorus like has a lower gyromagnetic ratio compared to fluorine, so you can only measure a shorter distance. I don't know, would you like, what do you think is a constraint on the H31 phosphorus distance if you try to apply a similar technique?
What are the constraints? The main constraint is hardware. If you can have a phosphorus fluorine probe, it'll be great. I can put fluorine on a protein and I can have a phosphorus, containing ligand. And, I would say, this kind of combination has been done by Jake Schaefer many years ago when he had that quadruple resonance probe, right? Transmission line probe. So the sky's the limit. I mean, I think absolutely I don't think there's any phosphorus chemical shift anisotropy is less than fluorine. So I think there's no problem applying that, yeah.
Okay, so thanks. This next question from Sang-Ho Park. How do you optimize your sample conditions such that protein to lipid ratio? What is the lipids phase at your experimental conditions?
Okay, so sorry, I didn't say anything about that. So we did these experiments in DMPC bilayers, and actually Katie's lab did a lot of DMPC, DHPC bicell measurements. And so it was just natural to go to DMPC. And the chemical shifts between the solid state that we measure, we assigned, 70% or something like that. And she has the solution NMR chemical shift. And we have comparison that they line up really well. So at 285 Kelvin, I think that's the effective sample temperature. So the sample is slightly into the gel phase of the MPC. If you go too high, everything is moving, you can't measure distances. So the optimization, these samples, protein lipid ratio is 1:25, molar ratio, and the line width is really good. We also have done experiments at 1:50. So in this case, because we have solution NMR data to compare with, we can really check, validate whether there's anything funny, right? So, and it checks out, it's really consistent. So it's nice to see, yeah.
Okay, we have a few more questions for anonymous attendee. So the first one is, in case of multiple 19F label sites in protein like CF, CF3, CF2, etc., could 19F nuclei would suffer from dipolar truncation to obtain distances?
Yeah, that's a good question. Yes, it can suffer from dipolar truncation. So we looked into this in RFDR, fluorine distance measuring, when we looked at that method. And so indeed, if you have nearby, fluorine competing with the distance you want to measure, that can be an issue. You can simulate a lot to account for that, but usually you would worry about, okay, do I really know the fluorine tensor, chemical shift tensor orientation? All of these things come in. So it is a bit tricky. So I would steer you to our JPC paper in 2019 on this dipolar truncation tensor, kind of chemical shift tensor, how that, introduces some uncertainty to the distance analysis and so on and so forth, yeah. So this could be an issue, but it actually is manageable. It's more manageable than carbon-carbon spin diffusion. Yeah.
Okay, can you explain why 2D peak intensities increase with longer redox mixing?
Okay. Sorry. So didn't explain that clearly. So what happens is, as you increase the redox mixing time, your D-phase experiment S, which I don't show, intensity comes down because the carbon signal decrease, right? It's dephased by fluorine. And then when you subtract to get the difference, then that intensity is high. And so this is basically a 1D version, 2D version of the 1D redox that you might be familiar with. In the 1D delta S, the intensity will increase as you increase the mixing time. So you progressively see spins that are further and further away from the fluorine dephaser. Yeah. So that's the idea.
Okay, how do you how do the distance you measure with 19F to protein compare to those measured with proton-proton using RFDR, for example, under fast MAS?
Okay, that's we haven't directly compared proton-proton and proton-fluorine in our distances in our system. What's special thing about this kind of fluorine-based strategy is you usually have just a very small number of fluorine probes, and they're just there can be just one fluorine in your sample, or in this case a drug that has 4 equivalent copies of fluorine. Okay, there are 4 fluorines. They're chemically equivalent but not magnetic equivalent. So then all your observables, whether they're proton or carbon, they get dephased by this very few fluorine spins, and that's very different from the proton-proton, right? And so when I say we would like to measure long distances, we're really going after long nanometer kind of distances. Of course, we also have the short distances, but the proton or carbon nitrogen-based experiments is focusing on short distances. When we say they're long range, we mean they come from residues that are far in the primary sequence, but not far in a spatial sense. And here we're getting spatially long distance. So there's a difference in meaning, right? So we think that both short and long are useful. And if you can get out to 2 nanometer, then we don't need to always think about fluorescence or EPR, right? So you can do a NMR measurement with very less perturbing fluorine probes compared to spin labels or fluorescent labels, right? So yeah.
Okay, CBS a question. He asked that, is it worth studying isotopically labeled polycationic antibiotics? Many of these novel antibiotics are peptidic in nature and shouldn't be too difficult to synthesize. And TPP is quite different in this respect. Yeah,
sure. There are other drugs, and I don't see the I can't read, it's out of my window. But anyways, sure. It just happens in this case. There's a TPP, and it's well studied for this particular protein. But if you have other type of drugs, peptide drugs or whatever, I think the thing is with fluorine label, it's usually orthogonal to whatever else you're doing, and that orthogonal labeling is the power of this approach. It's just, yeah.
Okay, what kind of 19F decoupling do you use during proton detection? Would spinal work?
Oh, yeah, when we spin less than 30 kilohertz, then we hit it with spinal TPPM to 100 something, 50 kilohertz or so. When we spin up to 40 or 50, then we dial back down to, watts with 10 kilohertz or something like that. Yeah, so we switch the regime. Can I make use of the large CSA of F19? Absolutely, yes. Yeah, not specifically necessary in the context of distance measurement, but in sensing conformational changes of things. Yeah, absolutely. This is very useful. Now, for distance measurement, CF3 group is really nice. You want all three fluorines to work together to dephase your remote whatever spin you're observing. So we like that. We don't avoid CF3s.
Yep. Russell has a question like, do you see any intermolecular contributions to the redox curve?
Oh, that's a good question. Yeah. In this case, in a membrane diluted by the lipids, there's no reason to worry about it. But you're right, in the small model protein like GB1, when we haven't done anything to dilute, I'm sure there's a little bit of contamination from intermolecular sources. That's why, that previous, this calibration curve is not quite spot on along that diagonal. And, I'm sure if we spend more time, diluting this GB1, we'll get, better accuracy and all of that. Yeah, you're right. So you could pay a little bit of price for seeing too far. But I think for any real applications, I don't think we'll run into that problem yet. If you're studying a membrane receptor like GPCR, its dimension is going to be large enough to make this not too much of an issue. Yeah.
Okay, last question. Could using different 19F configurations on different positions on 19F on the aromatic ring yield more distance restraints?
So that's an interesting question. We haven't quite looked at multiple fluorinated, so each aromatic ring with multiple fluorine, you can get these commercially. Would that help? Maybe. I think probably it won't hurt. If you don't care too much about, extremely precise single spin pair, and you just want to say, okay, I have this, small molecule that's lit up by fluorine, a bunch of fluorine, then sure, you can, go from that fluorine ball to some remote spins. Yeah, but it won't be a very, precise kind of analysis. Yeah.
Okay, thank you very much. Mei, for your very beautiful talk and showing us 19F reader and also like on
So
the...
Kong, I have a question before you.
Okay.
I have another one. If you
Okay.
Go back to your slides, Mei. You showed one slide right at the beginning of the fluorine at like 245 Kelvin or something.
Yep.
It looks somewhat, it wasn't rigid probably, but
Oh, this is the EMRE data.
yeah. I mean, you've got 4 fluorines there.
Yes. Yes.
And in principle, you should be able to get distances from each of those to the protein if you stop the motion. Right?
Yes, that's right.
So I'm just wondering okay, so that's 245. I wonder why that's so much broader
Yeah.
than the, it's 10 kilohertz. I guess you were decoupling.
So
Must have been decoupling.
right, this is a very complicated and subtle set of data. So the slow spinning, if you look at the 285, you might think, oh, everything is now moving. It's not quite true. There is a small isotropic component that comes from, we think, the drug. A little bit of it is in the solution. And that's only about 2% or so. And then if you look at the broad component here, their sidebands, if you track them, they're still not that much smaller CSA compared to 245. So when we fit those sidebands, like for example, this minus 110 ppm component, it is not bad. So now if you say, well, what's the rigid limit CSA? When we get to something like 50 ppm delta, the anisotropy parameter, then we think it's pretty rigid. I mean, okay, I don't remember whether we did a model just TPP by itself, a powder, we may not quite have. But anyways, we're pretty sure it's close to being rigid. The fact that you can pull, I think maybe a subtext of a question is, what are these different things, right? I think, so this drug it is hydrophobic enough. Part of it, or some of it goes into the lipid bilayer. And so we think this component, this orange component, intensity drops down. So it's not too close to the protein. And then if you look at the other deconvolution peaks, they all have different intensity contributions. So it's not like, oh, I have four corners of fluorine in this drug. Each corner is a different environment. That's not true. Yeah, so
Well, I mean, at a low temperature, it should be, though.
At low temperature, it is. It's a single CSA. But at this, the isotropic.
No, not necessarily. If it's hydrogen bonded differently, it could be. I mean, so that this sort of the question I'm asking is, if you get to lower temperatures, do you start to see any resolution in the fluorine peaks? And maybe you didn't go low enough yet. 245 might not be that low, but the broadening you see might be due to, to different fluorines coming out, starting to show up.
And
then at 308, there are two extra small peaks.
This is from residual starting reagent in the synthesis of the F4-TPP. So we checked that by solution NMR. I don't remember what this is, but I think it's, anyways, a little bit of leftover. We can try at lower temperature. I think it's unlikely that we can start to resolve, but you're right. If we could resolve multiple components, then that'll be wonderful, you
just need lower temperatures probably.
Yeah, well, if you can share your 500 cryogenic temperature, I'll be happy to put our sample on your magnet.
Okay, well, we've got to get it tuned to fluorine and get the fluorine cables out of the probe too.
Yeah, sure.
And there's fluorine all over the probe. That's a problem. I mean.
Yeah, that's true.
I mean, yeah.
Yes.
Okay, all right. So we should probably go on to the next talk. Thank you, Mei, for a very interesting presentation. And our next speaker is the famous Francesca Marassi from the Burnham Institute in La Jolla. And Francesca was an undergraduate chemistry major at the University of Toronto. She did her PhD also at the University of Toronto with Peter McDonald, and then she was a postdoc at the University of Pennsylvania with Stan Opella. And after a short stay at the Wistar Institute, she moved to the Sanford Burnham Presbyterian Institute in La Jolla, where she is professor, and now she's also director of the NCI Designated Cancer Center program for structures and molecules. Okay, so Francesca, the virtual floor is yours.
Thank you, Bob, and thank you, Kong, for organizing this excellent series. And I'm really honored to be closing it off here. I'll try and tell you a story about
finishing 2020. We're going to start it again in 2021.
Yeah, that's right. For this year. So I'll tell you about our efforts in understanding the way in which microbial pathogens interact with the host. We've been interested for some time in a protein called attachment invasion locus, or AIL for short, that sits on the surface of the gram-negative bacterium Yersinia pestis, this devastating organism that has been responsible for launching massive pandemics and devastation throughout history. And the motivations for looking at this organism and this protein are twofold. One is the potential of engineering organisms with antibacterial antibiotic resistance and disseminating them in aerolized form. And two is really as a way of understanding the way in which such bacteria can interact with the innate immune system. That is like the first line defenses that they encounter when they infect the bloodstream. And so it turns out that microbial pathogens like Y. pestis bind a repertoire of host extracellular proteins when they invade and infect. They colonize, pestis especially colonizes the lymph nodes, and it has three principal functions that are associated with AIL. One is the suppression. Let's see if I can pull up the pointer here. One is the suppression of the complement system. Oh shoot! Sorry, I'm trying to pull up the highlighter here. See if I can do it. Okay, sorry about that. Okay, so one is the ability to suppress the complement system, and that is the innate immune system of the human host, and that leads to survival. The other one is to promote the degradation of blood clots, and that is enhanced fibrinolysis, and this basically generates a more permissive pathway for the bacteria to infect. And the other one is to promote host cell adhesion, and that downregulates phagocytosis. And all of these activities really have this this effect of promoting the survival of the bacteria in tissue and in blood, and this enhances dissemination and proliferation, and leads to death. Okay, and so we've been interested in understanding this problem from the point of view of structural biology. And of course, I don't have to tell you that NMR is the ideal technique for looking at these kinds of systems. And with solution in solid-state NMR, we really have this range of molecular lengths and time scales that are available to us. It really allow us to look at things from very small molecules. Here's a cholesterol molecule to why can I bring up the, sorry there. Okay, to proteins, membrane proteins, if you're interested in them, I mean, soluble proteins, obviously, but proteins that are embedded in lipid bilayers or nanodiscs using solution NMR. And obviously with solid-state NMR, we have the ability to look at systems that are much more complex, okay? And so I won't be talking about these kinds of systems today. These are aggregates that form in macular degeneration deposits that are very interesting. There are spherules that have a lipid core, a shell of hydroxyapatite, and a layer of protein. But then you can also look at things like native bacterial cell membranes or native mammalian cell membranes, and ideally cells. And there's beautiful work now being done in this area of in situ NMR by many laboratories around the world. So our first efforts in solid-state NMR of AIL really focused on detergent-free phospholipid bilayers. And the reason for that is that early on we found out that detergents really wreak havoc on the function of this protein. They completely abolish ligand binding ability. And so what I'm showing you here are overlays of solution NMR spectra shown in black. Solution NMR spectra shown in red that were acquired for the protein embedded in nanodiscs. These lipid patches or lipid membrane patches that are encircled by copies of the apolipoprotein. And overlaid on those are solid-state NMR spectra from the protein embedded in liposomes, shown right here. Okay, 45 degrees, 10 degrees, solution NMR, solid-state NMR. You can see that there's a beautiful correspondence with perhaps a few changes, but illustrating the fact that the structure is unaltered, is the same. The beta barrel structure of the protein is maintained in both systems. The CP-HSQC spectra here were acquired with proton detection, and you can see the high level of resolution that we can obtain with optimized samples, duration, and careful optimization of the spectroscopic conditions. So using these kinds of systems, I won't show you all the data, but you can look it up in the work that we have published. We have been able to determine the three-dimensional backbone structure of AIL in both types of systems. And I'd like to mention that the structure calculations were really aided by this implicit solvent potential that we have developed as part of the Explore NIH package. And it's useful for soluble proteins because it increases the precision, the accuracy, and the quality of the structures. But it's also very useful for membrane proteins because it really promotes the correct membrane insertion of a protein, as you can see right here. And in cases where you might have highly dynamic loops, such as we see for ale, you have this correct exposure of the loops into the aqueous layer as opposed to them folding back onto the walls of the beta barrel. So it turns out that the loops are really where the functionality is, okay? Solution NMR is not really an option for going on with these types of experiments because what we really want to do is look at the interaction of the protein through the loops with proteins of the human host, and particularly vitronectin, which we have just identified as a major ligand. We have the structure of vitronectin, we have the structure of ale, but what we really want to see is the complex, and that becomes the complex in a membrane in a detergent-free system, and that becomes too large for solution NMR experiments. So also note the loops appear, and I say appear, highly dynamic with NMR, and it's not clear whether this is actually the case or whether this is an effect of using a highly artificial lipid bilayer system, in this case DMPC and DMPG with nothing else. And the reason for thinking that is that the bacterial outer membrane in gram-negative bacteria is actually much more complex than in a plain DMPC, DMPG. It's the outer leaflet is decorated with lipopolysaccharide, which the structure is shown right here. So there's a lipid A core with typically 6 acyl chains, and then an inner core and outer core of sugars. The presence of an O antigen or this extended sugar chains varies depending on the organism, and it turns out that in Yersinia pestis is limited to a small outer core of the protein. But suffice it to say that the protein sits in this lipid bilayer where the inner leaflet is phospholipid, the outer leaflet is primarily LPS. And in the case of Yersinia pestis, it turns out that the proteins and the outer membrane structure are highly synchronized. And what I mean by that is that upon infection at 37 degrees, so human infection temperature, the production of ale is highly upregulated. So it turns out to be about 30% of the outer membrane proteome. And the LPS acylation genes, that is the genes that are responsible for putting on these acyl chains, are downregulated. So that the only form that you get is this tetra-acylated form of LPS, not the typical hexa. And the core is limited to this inner core. Okay, so the two are really synergized at the human infection temperature. So things seem to be really optimized for infecting human. So we were interested in seeing what kind of effect this LPS might have on the protein structure in the loops. And some time ago, we did solid-state NMR experiments by supplementing the phospholipid bilayer, the liposomes, with the addition of basically lipid A from E. coli, which has a very, very limited outer core. And what we noticed through solid-state NMR experiments was that there appears to be an enhancement of conformational order, particularly of the loops. So we noticed this by noticing that the cross-polarization efficiency is enhanced. So you get this enhanced signal intensity in the presence of LPS, and also through measurements of T2 relaxation. Okay, so we noticed that the T2 of ale increases significantly in the presence of lipid A, from a value of about 7.4 milliseconds to 14 milliseconds in liposomes. And so these really indicate that LPS has an effect, has an ordering and rigidifying effect on the loops. Now, more recently, we've been performing all-atom molecular dynamic simulations, and these seem to bear out the experimental results. And basically, what I'm showing you here is the experiments, the MD simulation experiments performed in a bilayer where now the LPS is the same structure as the Yersinia LPS compared to a plain phospholipid bilayer or DMPC, DMPG, just like our experimental conditions. And you can see that the dynamics of the loops reflected in the root mean square fluctuations vary significantly with much, this is actually switched around. Oh, yeah, this is switched around, I'm sorry. With much dampened dynamics in the case of the Yersinia pestis membrane. And you can see that these loops, especially 2 and 3, stick out of the membrane and reach out to the surface where they presumably go and interact with their partners. So Chandan Singh in the lab at that time prepared nanodiscs with Yersinia pestis LPS, which we obtained from Andrei Anisimov. So this is actual tetraacylated LPS. And he was able to make very homogeneous preparations by replacing 6 phospholipids with 1 LPS molecule by taking care of the relative surface area ratios of the two. And the spectra, the solution NMR spectra in this case that he obtained, allowed us to compare the plus or minus LPS and to identify binding sites, specific binding sites of LPS on the protein. So you can see that we can map perturbations up into the loops, and we can also map perturbations off the sides of the beta barrel walls. So it turns out that this actually allowed us to identify 2 LPS recognition sites, 1 and 2, that I'm showing you here on each side of the sort of squashed beta barrel. And based on the structural information that we obtained, we were then able to mutate some of these sites and show that without going into the details of the gel, that they are important for preventing things like cell leakage, for conferring resistance to the antibiotic polymyxin in this case, and for promoting resistance to serum, okay, to all of the various complement and other factors that that are important for innate immunity in serum. So with this, the model that emerges is one where the cooperative functionality of AL and LPS is something like a mutually assured construction. Okay, so LPS is important for ordering the loops of AL, and AL itself is important for ordering the LPS outer membrane. Okay, and all of these are important for function. Okay, so this work really led us to realize that working in a native environment is absolutely important. And so we've been moving towards in situ NMR experiments in which ideally we would be in a cell. We're not there yet, but we are extracting cell envelopes from bacteria for NMR spectroscopy. So James Kent in the laboratory has been developing protocols for expressing AL natively in the outer membrane of bacteria, E. coli in this case, isolating these for Solid-State NMR in situ. And what he's working with is a gene now where instead of purifying AIL and then reconstituting it into phospholipid bilayers, he's attaching a PAL-B signal, that's a pectate lysate signal sequence, to the N-terminus of AL. And that takes advantage then of the inner membrane transport machinery and then the beta barrel assembly machinery for translation and then final incorporation of the protein within the outer membrane. And the results are really encouraging from these initial studies. So the protocol is to grow E. coli cells, lyse them, ultracentrifuge as you normally would, and then either take the E. coli cell envelope directly for NMR or use a detergent protocol followed by ultracentrifugation to take only the outer membrane for solid-state NMR. Okay, so the problem here, as many of you know, is that solid-state NMR, any NMR study really requires isotopically labeled biomolecules. We all know that. In addition, for proteins that are being expressed in cell membranes, you need to have targeted isotopic labeling. And this is really critical to suppress any background NMR signal that you might have from endogenous cellular components. Also, of course, the inherently low sensitivity of NMR necessitates that samples are enriched in the target protein. So these properties or these requirements are compatible with the properties of ale, which naturally comprises more than 30% of the proteome, of the outer membrane proteome at the infection temperature. We used E. coli in this case. So E. coli has been used as a model for Yersinia pestis, and strains derived from BL21D3, which we all use in the laboratory, have a Ref type LPS, meaning that it lacks the extended sugar O antigen, and so it's somewhat similar to Yersinia pestis. So, okay, so here are some of the results. Okay, so we took the full cell envelope, which means that we're taking the this whole section of the cell membrane. Okay, and so the initial attempts were with C41D3, shown right here, and these cells promote folded protein insertion into the E. coli outer membrane because they have this mutation that weakens the production of T7 polymerase. So these cells have pretty high expression of correctly folded ale, and we can tell the difference between folded and unfolded because you have this shift in the SDS page migration pattern. But they also produce appreciable amounts of background signal. And so to avoid that, we went to this other cell strain, D3, which is lacking outer membrane proteins A, C, and F. And these are the major outer membrane proteins expressed in E. coli. And so in this case, we also saw high-level expression. The benefit of these cells is that you can use rifampicin, which is an antibiotic that has been shown to suppress. It inhibits E. coli RNA polymerase, and it suppresses endogenous protein production. And actually, it was first used by Fabio Almeida to facilitate in-cell solution NMR studies of proteins. It's also being used by the Baldos Laboratory and many others since then. So these allow the use of rifampicin. So in the presence of rifampicin, you can see that we suppress background, but we still got a substantial amount of the unfolded ale protein. And so that was not suitable for our studies. So the final cell line that we tested was this LIMO21D3. And in this case, the T7 RNA polymerase can be controlled by inhibition of T7 lysozyme, and also rifampicin can be used to suppress endogenous protein production. And so in the end, I'll just direct you to these final lanes, the cell envelope and the outer membrane. You can see that especially in the outer membrane, you have these very clean preparations that essentially consist of folded AIL, okay, with some additional bands. So we avoid labeling of these other endogenous proteins by synchronizing the addition of rifampicin with protein induction, with ale induction, and with transfer to label media, so that when we do sale, it's primarily the only species that gets isotopically labeled. Okay, so as a baseline, we use these spectra, which are in liposomes of AIL that was purified and then reconstituted into lipid bilayers. You see here very nice NCA and CP, HSQC, H, and HSQCs, and many of these have been assigned, so they provide a very nice background for the work. And we found that when we then did the same kind of spectroscopy for the purified E. coli cell envelopes, we also got very nice spectra. So you know that these are essentially the first trials with this type of system, and so I find this highly encouraging. Many of these can be assigned just by basic comparison to the spectra that we obtained in liposomes. And in addition, we see peaks, some of which were seen in the nanodisc spectra, and some of which are new. So we'll have to go and assign. In the case of the proton nitrogen CP HSQC, it's good to note that the spectrum from liposomes was obtained on a sample that was per-deuterated. So it's per-deuterated ale inserted into liposomes. In the case of the E. coli cell envelope, that's not so easy to do. So what we tried to do in this case was to initially grow the bacteria in D2O to try and suppress actually the background. But the protein itself is protonated, and anything that grows after that induction period is also protonated. And so you can see that even with protonation, the quality of the spectrum is quite encouraging. Okay, and the fact that we can begin to see additional signals that we can assign to some of the loop regions is really what we want. Okay, so we want to be able to detect these loop signals like glycine 60 out here and 66, so that we can eventually map the interactions with the host proteins. And you can see here that we've mapped whatever we could pick out and assign perturbations. Interestingly, there are also some perturbations towards the end, reminding us that it's not just the LPS that is on the outer membrane leaflet, but it really is the entire membrane that's different. And we should not forget that there's a peptidoglycan layer underneath this, and so potentially also connections down there. We have started to acquire spectra, carbon-carbon correlation spectra. This happens to be PDSD spectrum, again again a sample in liposomes in red with a sample from E. coli cell envelope. And again, we can pick out many of the signals that we had assigned before. In addition to that, we see signals here circled in the gold or yellow box that are specific to the cell envelope components and not AIL. And so things like LPS, lipids, and peptidoglycan. And we are in the process of obtaining additional assignments. And finally, we've been collaborating with Galia Debelushina at UCSD to do some DMP on these samples. So this is the first DMP data set that we obtained. You can see that microwaves off, microwave on. We get an enhancement of about 28. The 2D, the first 2D spectrum is shown right here. Again, compare on and off. Of course, it's very broad because of the low temperature reduction that really freezes out all of the motions. But again, very promising. And one of the things that we're really interested in looking at are things like arginine side chains and the way in which they reach out and interact with the lipopolysaccharide. And so you can see here that we begin to, because it freezes out these motions, I think, we begin to be able to pick out the various arginine NECA signals in this kind of spectrum. Okay, so a lot of work to do, but I think very, very encouraging data. So finally, again, so what we care about, I want to cycle it back to, we care about understanding how this interacts with serum and the various human components. And so one of the interesting things about this model system that we developed is the fact that it begins to exhibit several of the pathogenic phenotypes that are observed in Yersinia pestis. So for example, normally the E. coli that you use in the laboratory can be killed if you expose it to human serum. So if we take bacteria that do not express ale, expose them to serum, they die completely. They cannot be plated. The control is shown here, where you inactivate the serum, then the bacteria can grow. So in the case where we're expressing AIL now, you can see that ale itself confers resistance. So exposure to serum now allows the bacteria to survive and to really combat the innate immune system that comes with serum. Okay. So the other property they have is that in the absence of ale, they do not form biofilms, they do not form pellicles, but in the Ale plus bacteria that we engineered, you see this very dense pellicle formation. So this is a crystal violet staining. Unfortunately, it's black and white. But what it means is that it's really the precursor of biofilm formation, one of the hallmarks of Yersinia pestis infection. And finally, we see that the bacteria that express ale are able to pull down this protein vitronectin, this major blood protein vitronectin from blood, both when they interact with NHS or normal human serum. Here's vitronectin being pulled down. The same does not happen when we inactivate the serum, nothing here. When we incubate it with pure vitronectin, plus ale pulls down vitronectin. And also when we incubate it with this domain of vitronectin, which we can produce in the laboratory, the so-called hemopexin-like domain of vitronectin. So what we did was we took our cell envelopes and we incubated them with human serum, which you can buy from Sigma or you can donate, and put them back into the NMR rotor. And in this case, data are very preliminary, but you can begin to map differences, chemical shift perturbations in some sites that happen to be very interesting, such as glycine 105 and G60, which are exposed to the extracellular surface, as well as sites down into the beta barrel. We don't know the meaning of that. We don't know what that means. I should say that this is human serum, so it contains multiple components that could be interacting with the outer membrane in addition to vitronectin. And so obviously we have a lot of work to do in this area, but the fact that we can now begin to probe this kind of system in a natural bacterial environment, I think is very exciting. And we're working with our collaborator, Greg Plano, to actually do these things in actual Yersinia pestis, which we ourselves cannot handle in the lab, but they can. Okay, so what's next? Like I said, what we really want to know is how it binds vitronectin. Here's the hemopexin domain. We know it binds. We want to get at that interaction. We have some preliminary data showing, these are solid-state NMR data, where now vitronectin is nitrogen carbon label and is associating with ale liposomes. So when we compare this spectrum, solid-state NMR spectrum in green, to the solution NMR spectrum of vitronectin in red, you can see many similarities, and we've been busy mapping the interactions in the presence of calcium, absence of calcium. We see interesting chemical shift perturbations in this area. We want to know how ale interacts with the other bacterial cell components, such as LPS, peptidoglycan, and other outer membrane proteins. And we are busy, for example, producing PLA, the plasminogen activator beta barrel that interacts with it. And the dream is really to reconstitute this ale vitronectin complex in situ for NMR spectroscopy. Okay, so with that, I will close. And I've talked about the people who worked on this project throughout the talk. Many people collaborated with us, so grateful to them. And I'll take questions.
Okay, thank you. Trying to get my video back on. It won't turn. Oh, there we go. Okay. So thank you very much, Francesca. And we have a couple of questions straight off. One is from Chun Boon. He wants to know, to clarify, is the SDH page or native page that showed the difference in unfolded and folded AL migration? All right. Maybe you can see the question yourself. All right.
Let me see if I can go. I think maybe you're talking about this gel right here. Maybe I can go and share again. So the question is, how can you tell the difference? So the difference is that, so it's a property of most of these beta barrel membrane proteins, and it's something that's called He modifiability. And that has to do with the fact that when they're folded, they migrate differently than when they're unfolded on SDS page. And so it's very easy to just run them on SDS page and see whether you have folded or unfolded protein in your membrane or in your preparations.
Okay, and Tim Cross has a question. How many arginine residues are there? And are they only on the phospholipid side of the protein?
Right. So I want to say that there's 6 or 7 overall, and there's 2 that are that face the interior of the beta barrel, whereas the other ones are engaged in sort of grabbing onto the LPS. And so we reported from our studies in the nanodiscs, we could see specific shifts in the arginine side chains due to the presence of LPS. And we're hoping that with solid-state NMR in the native membrane, we'll be able to see, to track some of these differences and understand really what's binding and what's not.
Okay, there was another question that popped up from Walt and then disappeared. All right.
Yeah, I can see. Like maybe I can ask the question. So like E. coli with AL inserted sounds like it might be particularly pathogenic. What do you do to limit environmental exposure?
I know. So this has come up. So we're not there yet. Okay. So Yersinia pestis has additional components that make it extremely lethal. So we're careful. I know that doesn't sound very reassuring, but we're nowhere near a highly pathogenic organism such as Yersinia pestis. I think it's interesting that we have a model system that lets us look at these various phenotypes, but that's not a danger yet.
Okay, and if you go back to, could you go back to your DNP slide with Galia?
Yes, I can.
I mean, you have a black spectrum over a green spectrum. Yeah. Is the black spectrum room temperature, or is that low temperature?
No, the black spectrum is this spectrum. Okay, the NCA spectrum right here. So that's taken at 10 degrees Celsius.
Oh, okay. All right. Can you go back to the
so there is a temperature difference, yes.
Yeah. So you're seeing a lot of extra cross peaks, or many extra cross peaks here. Yeah.
Yeah, I mean, and I think in part, so one of the problems, let's say, or the challenges with the system is that the loops really, they are dynamic, and they undergo this, intermediate scale dynamics that makes them invisible. And so I think what happens here is that we're freezing out all the motions. And so you start to see, not only are they broader because of that, because potentially you're capturing all these different conformational states, but also because I think we're seeing additional signals from motion studies
Yeah,
out.
it seems very clear in the arginine region.
Sorry?
It seems very clear up in the
Yes, that's
arginine
right. Yeah,
region.
where you start to count. That's right. So there you start to count these additional.
Yeah, and also the lysine.
And the lysine too.
Yeah.
That's right. The lysine that folds in is very clear too. So I think, I mean, it would be nice to, to go back. We've learned a lot since then on how to prepare these samples and obviously collect additional data and do this more carefully.
The other thing that you might consider is that Anne Lesage, for example, has shown that when you it's been sort of assumed that you're seeing inhomogeneous broadening, chemical shift broadening when you go to low temperatures. And Anne has
Right.
shown pretty definitively, and there have been other cases as well, that indicate it's low temperatures, there's a lot of homogeneous as well as inhomogeneous broadening. And the way you get rid of that, I think Galia probably has a 3.2 millimeter probe that won't spin more, maybe about 10 kilohertz or so. But if you use a 1.3 and go up to like 40 kilohertz, which you can do with nitrogen, you really do improve the resolution a lot. And also, of course, higher fields too.
Yeah, well, that would be really great to try because, again, like I am pretty encouraged by these preparations. I think, we are labeling ale at the expense of all the other stuff. And, the structure is clearly folded. And so it would be wonderful to try and get high resolution.
So Galia says she has a 1.9 millimeter probe, and how fast can it spin? At 90 K, at the
Galia can answer that.
well, maybe she can answer. She seems to be online.
Yeah, she's online. Yeah, 25.
Well, that's probably not quite fast enough. It helps, but it's you need to go probably to 40. 60 would be even better, of course. But Galia, do you want to unmute yourself?
Hi, can you hear me?
Yes.
Yeah. So, I mean, these were actually acquired at 12 kilohertz, but in the 1.9 millimeter probe. So we can certainly try, and we can try going faster. And yeah, I don't know if it will be enough, but that's what we have.
Yeah, well, I mean, it's the higher spinning frequencies and the higher magnetic fields are, probably the way to recover a lot of the resolution that you see at the higher temperatures, room temperature, and so forth. Yeah.
Yep.
Yeah, certainly a lot to do.
There's one final question. Could you reconstituted isotopically labeled ale into E. coli? This might avoid having to grow and force them to express it.
Yeah, I honestly cannot think of a way of doing that.
Mm-hmm.
I mean, you have to produce it, purify it, so it's refolded in detergent, then detergent is dialyzed out. I don't know. I have little hope that would work, actually. But I think that the data that we have shown indicate that this is actually not a problem. And, especially, I think also these results, now it's true, if you look at the HN CPHSQC, I mean, that's not deuterated. Okay, so that's at 900 megahertz with 1.3 millimeter probe spinning at not quite 60 kilohertz, but close to it. So I think that itself is also very, very encouraging, just because it's not deuterated. So I think that, we could do some work on sample preparation and isotopic labeling schemes. And
that's very nice spectrum.
Yeah, I think that they are very nice. I mean, the spectrum in liposomes is beautiful. That's deuterated. And that's highly optimized. So I think, given that this is the first, it's the first one that we obtain. So I think we can do better.
Okay, so there don't seem to be any further questions. So I'd like to thank both Mei and Francesca for two really beautiful talks today. exploring different ways to approach membrane proteins, and that's an exciting new areas that we can all start to think about with solid-state NMR and DNP. And as Kong mentioned, if you didn't hear him at the first in his announcement, that we're going to take a holiday break, a Christmas, New Year's break. And so this will be the last of the ZOOMinar series for 2020, but we will restart the series on January 12th. Is it January? Yes, January 12th of 2021. All right. And so we're going to continue this for as long as it seems to be necessary, and it seems to be people attending it and providing and are interested in it. So again, we will start on January 12th, and we will be sharing the time every other week with the educational seminar on solid-state NMR and DNP. So again, Mei, thank you very much, and Francesca, thank you very much. And we hope to see everybody back here on, say, January 12th or so.
Thank you.
Bye, everybody. Happy New Year.
Bye.
Already.
Merry Christmas, Happy New Year, Happy Hanukkah, and we will see you in January. Okay.